multifunctionality, so it can work both as a diagnostic nanoparticle as well as a therapy-induced nanoparticle, because it is carrying both the drug and your quantum dot, so it can work as both. Then, of course, it gives you greater sensitivity in diagnosis, because you have quantum dots which are much more sensitive and much more brighter as compared to your regular conventional dye, and therefore they can detect the signals of cancer much earlier than your conventional dye can. At a lower concentration of the biomarker, you'll still be able to detect the signal of cancer and then also try and identify it and try and classify it. Targeted delivery, because your nanoparticle is having what is known as the ligand. This ligand could be an antibody or another protein that binds to a specific protein on your targeted cell. So therefore, now your docking of the nanoparticle is not to any other type of cell, but only to a specific type of cell. So therefore, you can target your drug only to the specific type of cell that you want to. And because most cancer drugs are cytotoxic, so now the cytotoxicity comes only to the cancer cell in question and not the other healthy cells of the body. So in that sense, this is more targeted, and there is less collateral damage. High therapeutic index, because now again the drug is getting targeted exactly to a specific cell, and also because you can control the release of the drug. The drug is there in the amounts required for lethality. It is not given in a sudden dose, because when you give it in a sudden dose, what happens is the cell will activate a pathway where the drug can be pushed out of the cell, and most of this happens via G-glycoprotein pathways. These are your transport pumps that preferentially remove the drug or clear the drug from the cell, and therefore the cell may not be then susceptible to cytotoxicity. To ensure that this does not happen, you have your, again, a sustained release. In sustained release, this pathway is not activated, and therefore the cell progressively moves into the cytotoxic pathway and dies away. And then, of course, nanoparticles can enable you non-drug therapies. Non-drug therapies meaning that apart from chemical drugs or your proteins or your RNA or your DNA that you give as a drug, you can also use nanoparticles as non-drugs. Non-drugs meaning there is nothing of a drug that you're giving. You're trying to kill the cell by inducing specific heat activity or specific pressure activity or a specific electromagnetism that allows you to, while your nanoparticle has a ligand, it docks to a cancerous cell. Then you can, this nanoparticle now heats up because of its interaction with the cancerous cell, and that heat generated is able to kill your actual targeted cell. So this is a non-drug therapy. It is known as photothermal therapy or photodynamic therapy. And then, of course, you can have combination therapy. So you can use a combination of drugs here. These combination of drugs could be, several drugs put together, because it is also known that, a single drug may not be cytotoxic enough. And what you want is actually several drugs put together. They will act in synergy and then kill the cell. So that is, again, very important. And therefore, combination therapies can be used. Combination of drugs can be used. A combination of drug and the photoactive particle, the quantum dot, can be used together. And many things can be done here so as to ensure that finally you have a higher effect. The actual target that you wanted to kill is actually killed and removed from the body. So while regular chemotherapy and radiotherapy are not so targeted, and therefore there is a lot of side effects that you have from cancer treatment. One of the most visible side effects that you can have from cancer treatment is when the people lose hair immediately. Even after the first chemotherapy, you will see people have lost a lot of hair. That is because the therapy is so strong. The radiation therapy or the chemotherapy is so strong that these hair follicle cells get killed, and therefore immediately or instantly there is a huge hair loss that happens. So you want to try and avoid these collateral damages, and therefore what you do is to use a combination therapy, use a targeted therapy, and you also want to diagnose well. Therefore you add a quantum dot or other fluorescent material so as to ensure that only the targeted cells are killed, right? So then we move on and we see how this targeted therapy works. If you see here, I show you two types of micelles. This is a micelle where you have a dye block, basically dye block polymer, which is having an amphipathic and hydrophilic and hydrophobic end. So therefore, you can put a drug in the center of this, and based on its alignment, it will align into the hydrophobic in the center and hydrophilic at the end. This missile can then move in the body. This missile is also loaded with a ligand on the top here. This ligand can bind to a specific receptor on the cell. This receptor is specific only to cancer cell, and therefore now your nanoparticle is only docking to the cancer cell and not the other cells of the body. Once the docking is done, now this nanoparticle will be internalized, and this internalization can happen by covalent mediated pathway or collateral mediated pathway or other pathways. Simple phagocytosis can also happen. So there are several ways by which the nanoparticle can be internalized, and largely it depends on the size and nature of the nanoparticle that you have, which we had done in previous classes. So then once it is internalized, if you see, there will be a drug that will be released, and this drug will then have its effect. Most commonly, the drug effect is to kill the cell. Let's say you're using a small inhibitory RNA, then the idea would be to control the expression of certain oncogenes. Oncogenes are the genes that promote cancer. So you would want to stop the expression of such genes that are promoting the cancerous behavior of the cell. So for that, you can use a small inhibitory RNAs, microRNAs, or you could also, if you're interested in genetic engineering, then you could also produce a nanoparticle carrying a DNA payload that can go into the nucleus and exchange with the wrong segment and give you the correct combination. So the ligand receptor interaction allows for unloading of the cargo, the nanoparticle protein corona composite. So remember, we talked of how the nanoparticle, when you introduce a nanoparticle in the bloodstream, the first reaction is to coat the nanoparticle with a set of proteins, right? And this set of proteins would form two layers on the nanoparticle, forming what is known as the protein corona. You will have a hard corona, or the inner layer, that is in direct contact with the nanoparticle. And then you will have a soft corona, which is the outer layer. And remember also that the protein corona is dynamic. Dynamic meaning the composition of proteins keep changing as we move in time. And I also told you that actually the physiological response of the cell is not to the nanoparticle, but to the composite that is formed, which is the protein corona nanoparticle composite. So the nanoparticle protein corona composite interacts with the cell surface and is finally internalized by diffusion, phagosomy, clathrin, or covalent mediated pathway, or endocytic or other endocytic pathways. The cargo is released after the digestion and lysis of the endosome. Therapeutic or optical properties of the cargo may be qualitatively and quantitatively analyzed, and that would help you see whether the drug is actually having an effect or not, right? And here at the bottom, I show you a lipid bilayer. So this is a liposome. Lipid bilayer, lipids, phospholipids are amphipathic. They have a hydrophilic head and a hydrophobic tail in between. So when you put them, they can naturally assemble together into what is known as liposomes. And liposomes would have the tails towards each other. So this is your hydrophobic portion of the liposome, and here you have a hydrophilic interface here and a hydrophilic interface here on the outside, right? And the drug can be accommodated. If the drug is hydrophobic, it can be accommodated at the center of the liposome, or it could be, if it is amphipathic, it could be accommodated in this membrane itself and can be aligned just like the lipids are. So therefore, both of these can be done. And this liposome is again carrying the ligand. So this ligand binds to a specific receptor on the target cell, and then of course it is internalized, and after internalized, again it will release the drug and show you the effect, right? Hydrophobic drugs are important because most of the cancer drugs are hydrophobic, so therefore you have to ensure that a hydrophobic drug is delivered to the cancerous cell. A lipid bilayer liposome will not be possible to ensure that a hydrophobic drug is released. So therefore you can use a monolayer liposome where you have the internal hydrophobic tails, and in those hydrophobic tails, along with that, you can have your hydrophobic drug. The majority of anti-cancer drugs currently are used or in clinical trials are hydrophobic, including, for example, paclitaxel, which is widely used for treating ovarian, breast, and non-small cell lung cancers. Hydrophobic drug molecules may not be soluble enough to cross the aqueous environment, and therefore they have to be packaged into a nanoparticle. One efficient approach to overcome the poor water solubility of hydrophobic drug is to encapsulate it in a nanoparticle-based carrier, right? The particle should have good loading capacity, and which can be easily met through the use of hydrophobic or amphiphilic material. So you can use hydrophobic material to make a nanoparticle, or you can use an amphiphilic material, which is which has one end that is hydrophobic and the other end that is hydrophilic, right? A variety of carriers have thus been developed for delivery of hydrophobic drugs, including those based on polymer micelles and polymer nanoparticles. And then, of course, it has been shown that a hydrophobic drug can be concentrated a lot more, 50,000 times more, when it is encapsulated in a liposome-based carrier as compared to when it is given naked. So the drug bioavailability is a lot more when it is loaded into a nanoparticle as compared to when it is given without the nanoparticle. Then, of course, once the nanoparticle has docked into the cancerous cell, it is important that the drug is released in a steady, controlled manner. It cannot be that immediately the drug is entirely released and then you'll have a problem. Therefore, there are methods of doing that. You can have a sustained drug release. And now what you also have are smart nanoparticles. Smart nanoparticles means they will respond to a stimuli. Response to stimuli means that, it is known that most cancerous tumors would have a slightly lower pH, as in around 6.5, 6.7, as compared to normal healthy tissue cells, which is around 7.4. Normal body pH is around alkaline, lower alkaline, not very high alkaline. But in tumor cells, if you'll see, the pH is slightly lower, around 6.7, 6.8. And that can work as a sensitive method of allowing the release of the drug in those cells, right? So that is one thing. You can do that. And then, of course, you can control with the help of temperature. If the temperature is below the threshold, the drug would remain concentrated. If the temperature is above the threshold, the drug can be released and come out on the surface and can interact with the cancerous cell. So those are methods of trying to have control over how the drug is released. Then, if you remember from DNA and DNA protein nanoparticles, we had done this. So here you have a nano vault. This is basically made of DNA origami sheets. And this DNA origami sheet is closed in closed conformation, and the closing is controlled by here what is known as the DNA lock. And if you remember, the DNA lock can be opened because this is more in the form of what is known as the toehold mediated strand replacement. So this yellow strand, when it binds to this DNA that is there present here, it can then allow it can progressively hybridize with the pink strand and replace the yellow strand, right? And once it replaces the yellow strand completely, the lock can be opened. When the lock opens, you have the exposure of the enzyme or the drug that you had in the center. And now this drug can impact its immediate surrounding. It could be a cell, it could be your blood fluid itself. Right? So encapsulation and controlled activity of enzyme in hybrid nanostructures is what has been achieved. This has been achieved at the experimental level, and we're working on how to actually implement it at the individual level, right? At the level of humans. So, many of the things in nanobiotechnology are still at the promising stage, still at the development stage. They are very promising in the sense that they have shown its impact in cell culture, but in actual body, the impact has to be still proven. And very few drugs have now finally crossed the stage where they can be, and they are shown as safe and can be used in humans. So nanotechnology is a fast developing field. Nanobiotechnology is all the more fast developing field. Many things are promising, many things are under development, right? So then if you see the sequestered enzyme, which is in this case happens to be bovine alpha chymotrypsin, is inaccessible to its substrate in the closed conformation. So this is a closed conformation. There is nothing that can happen. The enzyme is not exposed to the substrate and therefore no reaction happens. And then of course the double-stranded DNA locks that hold the vault closed are displaced through a comparative interaction with a complementary key, which is a single-stranded DNA. So if you're a single-stranded DNA which has complementary hybridization with this, will now start competing. Remember, this blue strand, which is the original strand, would have a bit of extra DNA sequence that is known as the toehold. So it will first hybridize through the toehold and then progressively keep hybridizing and replacing the blue strand. So the pink strand is the one that will have the toehold. Okay. The opening of the vault enables access to the enzyme conversion of the substrate to the product. Then, of course, we had talked of this example as well, where you have again a DNA lock here. This is a sheet, and here you have an enzyme. In this case, this is a thrombin that we have attached. And this lock can be opened by nucleolin, and it is specifically overexpressed in cancerous tissue, And therefore, as and when the nucleolin comes and binds to this DNA lock, the lock opens, exposing the thrombin, and thrombin will then cause lysis of the cells. So the nanovehicle consists of a DNA origami tube containing the blood clotting protein thrombin. The tube was designed to remain closed in the bloodstream until it encounters nucleolin overexpressed on cell surface, right? So this nucleolin is overexpressed on cancerous cell surface, so that can then work and bind here. Then the sheet would open, exposing the thrombin, and then that can cause the lysis. One strand in the lock is an aptamer for nucleolin, a DNA binding protein overexpressed in some cancers. Right? So one of the most potential ways of doing a non-drug cancer therapy is to basically induce heat or ferromagnetic response. And that heat or ferromagnetic response can allow certain enzymes to be expressed, and that would possibly take the cell to the apoptotic pathway. So here you have the nanoparticle. Nanoparticle is internalized by the cell into the lysosome here. And then, of course, the nanoparticle can be activated so that its magnetic field is switched on, right? So binding to the lysosomal membrane, disrupting the lysosomal membrane, and now the nanoparticle is free. The pH goes down, and this results in the basic lysis of the cell via the apoptotic pathway, right? Many preclinical studies have been performed to demonstrate that both radio and chemotherapy can be enhanced by simultaneously operating hypothermic therapy. So this works in combination with radio and chemotherapy. The hypothermic therapy, photothermal therapy, is based on the conversion of light energy, usually in the form of near-infrared region, into heat energy to induce subsequent cellular necrosis or apoptosis. Some current nanoparticle technologies for hypothermic therapy include ferromagnetic nanoparticles such as iron oxide, doped iron oxide, and supraparamagnetic iron oxide nanoparticles, SPION, as well as carbon nanotubes. So for phototherapy, you can use iron oxide and its derivatives and also carbon nanotubes. They can be controlled once they are docked into the cell. Their temperature can be increased, or they can also be, their magnetic properties can be activated. Once the magnetic properties are activated, this then results in the basic, the death of the cell via the apoptotic pathway. Now, gold nanoparticle, we had talked of this, that gold is used very commonly for biotechnology purposes, both for diagnostics as well as for therapy. And there are reasons for this, why gold is used. And if you remember, in an earlier class, we discussed that gold is highly biocompatible and can also be very easily bioconjugated with other biomolecules. So that gives gold this additional, utility, and it itself is not very reactive. So therefore, it is biocompatible, and it can be conjugated very easily with other biological molecules. So you can include your drug, you could include your targeting, antibody, and so on and so forth. That makes it more specific to use in cancer. They can be administered into the local tumor area while minimizing the non-specific distribution. So you can administer into the local tumor area by giving a local injection. They can be activated near infrared laser light, creating the ability to penetrate deep into the biological tissue. So near infrared light is what you require for deep penetration into the body tissue. And because gold can be activated by near infrared light, so even the cancer which is not on the body surface, which is somewhere deep into the tissue, can also be targeted. They can be modulated to create multifaceted cancer nanoparticle, for example, photothermal therapy or drug delivery. So the gold particle can be conjugated with your drug. It could also be alongside. It could also be conjugated with a ligand that binds to a specific receptor on the cell surface that you're targeting. So all this together makes gold a very viable option for cancer therapy. The efficacy of plasmonic gold nanoparticles for thermoablation of various cells types has been demonstrated in multiple studies. So thermal ablation means, again, you send the gold particle to a targeted cell, and then you enable the gold particle to generate heat. And when this heat is generated, the cell that is targeted gets killed. So thermal ablation with gold is also possible, and therefore gold nanoparticle is a very useful material for cancer therapies, both drug-mediated or non-drug mediated. Then, of course, there are several cancer drugs that are there, and if you can see, you have a lot of hydrophobic drugs, you have a lot of hydrophilic drugs, and then you have also highly charged. So these molecules are currently under checking. We are not sure whether they would work or not, but several of these small inhibitory RNAs are now undergoing trials so as to give a possible therapy to cancer, right? And most of the siRNA would work in terms of bringing down the expression of the targeted gene, right? So broadly speaking, cancer therapeutic agents can be classified into two major groups, hydrophobic and hydrophilic, depending on their aqueous solubility. Alternatively, they can be categorized as highly charged or neutral drugs based on their electrostatic properties. When choosing or designing a nanoparticle to be used as a carrier for a specific type of drug, it is of critical importance to know the properties and behavior of your drug itself, right? So as I said, if it is hydrophobic, then you cannot use a bilamellar lipid liposome as your delivery vehicle, right? In that case, it has to be a micelle or a unilamellar lipid liposome, right? So that is important, and that you have to know in order to achieve optimal encapsulation, efficiency, and the desired release profile. So you also have to ensure that the drug is released in a sustained manner. So not every nanoparticle will work as per your requirements. You'll have to then choose and pick from the available nanoparticles which one is the best suited for a specific type of sustained or continuous or controlled delivery of the drug. So there are a whole lot of drugs with nanoparticle based therapeutics that are under trial, and some that have already been approved for cancer treatment. So you can have a look at this list and remember at least one or two names, so that when you write your answer in a subjective answer, then you can substantiate your answer with examples. It is always good to write an answer with examples so that you can show to the examiner that you know what you're saying, right? So therefore examples are important up there. What are the challenges in the cancer treatment? So there are challenges because while you can do all the targeting and everything, the body itself has a lot of specifically dedicated cells that ensure that these dead cells or the remains of dead cells or the foreign particles that come in can be made to be targeted by the immune system. And this system is known as the reticulo endothelial system. So nanoparticles that are present in the body are largely taken up and eliminated by the reticulo endothelial system, a network of cells that are distributed throughout the body. So if you look at the reticulo endothelial system, you have microglial cells in the brain. These are part of the reticulo endothelial system. Then you have macrophages in the lung. Then you have tissue macrophages or histiocytes. Then you have Kupffer cells in the liver. Then you have glomerular mesangial cells in the kidney. Then you have macrophages which are produced in the spleen. Then you have osteoclasts in the bones. And then of course you have macrophages in the lymph nodes. So there is a complicated system of cells in the body which can work in a coordinated manner to ensure that any foreign particle or any other nanoparticle that has entered the body is targeted to the immune system, right? So therefore, the nanoparticle that you give to the body, the nano injection that you're giving, that nanoparticle has to skip this reticular endothelial system to be effective in drug delivery. Nanoparticles that are present in the body are largely taken up by and eliminated by the reticular endothelial system, a network of cells that are distributed throughout the body. The functions of reticular endothelial system is the inactivation and elimination of dead cells, bacteria, virus, and infiltrated small particles. So nanoparticles would come under the infiltrates. Infiltrates means something that has infiltrated into the body, which is not body's original, right? Reticuloendothelial system or mononuclear phagocyte system is a network of cells and tissues that are distributed over the whole body and are especially abundant in the blood, liver, spleen, lung, lymph nodes, bone marrow, but also in connective tissues and the brain. The cells are phagocytic and hydrolytic, right? So these cells would immediately phagocytize and hydrolyze your nanoparticle. This network of cells consequently possesses important functions of immune defense against the disease. So it is understood that these will be immune cells and they would try and, be the first, be a layer of defense against the invading pathogen or the nanoparticle. Without special surface alterations, more than 90% of the nanoparticles are recognized by reticular endothelial cells and taken up by these cells and then disposed and eliminated from the body. So unless you have a special coating around your nanoparticle howsoever, whatever size or whatever shape it has, it will not be able to reach its targeted cell because it will be in the way waylaid by reticular endothelial cells, and they would then immediately phagocytize, digest, and remove it from the circulation. Mostly the nanoparticles will be eliminated through urine and also through the liver, where they'll now be part of your gallbladder. That the bile, it will be eliminated in the bile, and bile is stored in gallbladder. So therefore they'll get restricted in the gallbladder. A part of it would also be cleared by the spleen, right? So for these reasons, particles for therapeutic purpose have to be specially designed, for instance, to achieve an enhanced tumor concentration, right? So the particles have to be specifically designed. They must be camouflaged well enough. And we talked yesterday of polyethylene glycolation and how that can help us in hiding the identity of the nanoparticle and not eliciting an immune response in terms of opsonization or in terms of the cells of the reticular endothelial system, right? So there has to be a reality check, and I give you a Nature reviews from 2016. Nanoparticles are designed to alter the biodistribution and pharmacokinetics of a small molecule drug or contrast agent in patients. So biodistribution, you understand how the drug is distributed across the body. What we want is a specific concentration of the drug on the targeted cell and not across the whole body. So therefore, we add a ligand so that once the cell comes across, once the nanoparticle comes across the targeted cell, it gets stuck with that and it doesn't move then across the elsewhere in the body. Pharmacokinetics is basically what the drug does to the body, right? So what the body does to the drug is what is pharmacokinetics. So if you see, when you take the drug, there are 4 things that happen. One that is known as ADME: absorption, distribution, metabolism, and excretion. So ADME features of your drug have to be worked out so as to ensure that there is enough drug reaching the targeted cell and in good concentration enough to be effective. So the therapeutic index of the drug would depend on its ADME features. ADME includes absorption. So if you're giving intravenously, then again it will move through the blood into specific tissues. So it has to be absorbed well to be actually able to reach the targeted tissue. Then distribution, it has to be distributed, and preferentially it should get enriched into the location where you want it to be enriched. Then of course metabolism, your drug should not be metabolized very fast, because then of course the drug will not show its effect because it is itself getting altered very fast. So the drug should not be metabolized too fast. If it is metabolized too fast, it will basically not be bioavailable for its action. And then excretion, of course, the drug should not stay in the body for too long, because if it stays for too long, then again it would be toxic to the body, and therefore it has to be ensured that the drug or its metabolite product that is present in the body is finally excreted effectively from the body, either through urine or in feces. So what you want is to improve the efficacy of the drug. So efficacy of the drug is measured in terms of its therapeutic index. Higher the therapeutic index, the better the drug is. A reduced systematic toxicity, so therefore you're targeting it to specific cells, and or offer better imaging signals. When you're using quantum dots or other fluorescent things, then it is also to offer you better imaging signals. These advantages have been dampened, lack of translation to patient care despite large investment. So what this report says is, while nanoparticles are very promising, actually, if you look at this, the actual targeted delivery, the rate of targeted delivery is very low, and it is not something that we can, as of now, be very happy about, or we cannot be cheerful about it, because there still is a lot that needs to be done so as to make the promise translate into reality, right? So the reality check must always be there. And nanoparticles and bio-nanotechnology is a developing field. There is a lot that has to be, it has to be customized, it has to be standardized so as to be useful to actual treatment cases, right? So as I showed you, there are a lot of drugs and nanoparticle mediated drug treatment that are under trial. Phase 1, phase 2, phase 3. There are 4 phases in a drug trial. And the 4th phase is where you have human subjects, where it is tried first. And then when you say that there is no effect, there is no side effect, then it is put to a larger population. And then you have what is known as post-market surveillance, where you again want to check what has happened to the, whether the reports of the drug are right or not, and whether it is causing any side effect or not. And there is an interesting example here. If you see, all of you must be knowing about Viagra. it is a sex stimulant. But when it was initially launched, it was not launched as a sex stimulant. It was launched as a heart drug. And what was found was that instead of heart, the effect is more seen in terms of the sex stimulation. So finally, it was redesigned to be launched as a sex stimulant drug based on post-market surveillance. But the idea is all of this that we have seen till now is promising, is likely to get translated, but as of now, the translation is not so efficient. And of course, if you remember class, we had also talked about how in cancer cell, the methylation pattern of DNA changes. So if you see here, this is a normal cell. Red indicates methylation and blue indicates unmethylated cytosine. Methylation on DNA happens on cytosine nucleotides, right? And if you see here in a normal cell, most of the repeat sequences are methylated and therefore they cannot move. The transposons, if you know, these are jumping genes, so they can move from one location in the genome to another. So if they're not controlled, they would keep jumping and they would again, they would break your genes and they would then cause a lot of damage. And therefore, what happens in normal conditions, most of the transposons are highly methylated so that they cannot move from their location to a new location. And if you see in the cancer cell, this methylation is removed and now it becomes blue. And therefore, now you have the cancer cells, the genome is a lot more unstable as compared to what you have in a normal cell. Then, of course, if you look at the CpG islands are regulatory regions which are present upstream, 5' upstream of the genes. And here again, if you see, they are unmethylated in normal cells and they undergo a certain amount of methylation in cancerous cells. So the pattern is absolutely contrasting if you see for normal and cancer cell. Likewise for CpG island shores are again unmethylated. Island shore is just outside the island. So these are unmethylated again in normal cells and highly methylated in cancerous cells. And then of course gene body, you have a bit of methylation, but gene body methylation means methylation of cytosines within the gene. And that is there in normal cells and it is again reverted in cancerous cells, right? So what happens is the idea is that when you look into the cancerous cell, the actual genome methylation patterns are very different between a normal cell and a cancer cell. And what people have now done is this can be used as a very clear diagnostic method to identify whether the cancer is setting or what is the extent of the cancer that is there. So now you can test cancer in a single cell with a naked eye through a color change. So what you do is you add your gold nanoparticle to the DNA, which could be normal or cancerous. And then, of course, within 5 minutes of the reaction, you'll be able to see unmethylated DNA in a different color. And you will be able to see cancer DNA in a different color and a normal DNA in a different color. So this is a colorimetric test for cancer, which is under development, highly promising, and likely to be the best as of now to predict whether the cell is taking a cancerous pathway or not. And this is based on only the DNA. And what it is based on? It is based on the methylation patterns in the DNA. The methylation patterns DNA I showed you are contrasting between normal and cancerous cell, and therefore this can be used as an early diagnostic test for cancer. This is a very promising technology that has come up only now in the last 1 or 2 years, and it is still being developed, and this will likely be the most important test that you have for a cancer diagnostics, right? Because it is very small, takes very less time. Now we just take a look at the timeline of cancer nanomedicine. So 1964, the liposome were first published. They were accomplished. Then, of course, sustained delivery of low molecular weight compounds using silicone polymer was done in 1964. 1976, the first controlled release polymer systematic for ionic molecule or macromolecules was developed. Then the first targeted liposome was developed. So you had stages of evolution. First, you made the liposome 64. Around 16 years later, you had a targeted liposome. So now you could put a drug in between, and this would then go and bind to a specific cell. Then, of course, you had the discovery of EPR as enhanced permeability or retention effect, right? Which is in the vasculature of your cancerous cells is slightly different, which allows for retention of larger molecules or larger size nanoparticles. So this is known as enhanced permeability or retention effect. Then, of course, you have long-chain nanoparticles, liposomal, first drug approved by FDA in 1995 that could be used as with a targeted liposome. And then, of course, you move on. The first targeted small inhibitory RNA drug that could be used with the targeted liposome, and so on and so forth. We'll discuss this in the next class.